Welcome to our lecture on developmental biology. Today, we are diving into one of the most fascinating topics in science, pattern formation. Have you ever wondered how a simple, round egg knows exactly how to build a complex body with a head, arms, and a heart in all the right places? It is not magic, but a highly orchestrated biological process. Let us uncover how this incredible transformation happens. To start, imagine you are a construction worker building a massive skyscraper. You can't just dump bricks and steel on a lot and hope a building appears. You need a highly detailed blueprint. In the biological world, a developing embryo faces the exact same challenge. Pattern formation is this biological blueprint. It tells a growing cluster of identical cells exactly where the head should go, where the arms should grow, and where the heart belongs. It is the process that transforms a simple, featureless egg into a beautifully organized, fully functional living creature. Now that we understand the blueprint analogy, let us look at the formal definition. In developmental biology, pattern formation is the process by which initially equivalent, identical cells in a developing tissue acquire distinct identities and spatial arrangements. Think of it as setting up the fundamental coordinates of the body. This process establishes the primary body axes. It defines the anterior-posterior axis, which runs from head to tail, and the dorsal-ventral axis, which runs from back to belly. Without this, cells would not know their place in the overall structure. So, how do cells actually receive these coordinates? At the cellular level, pattern formation is largely driven by chemical signals called morphogens. These morphogens are secreted from a specific source and spread through the surrounding tissues. As they spread, they create a concentration gradient. This means cells closer to the source receive a high dose of the signal, while cells further away receive a lower dose. By reading this local concentration, each cell determines its exact physical coordinates within the embryo and activates the specific genes required to become the correct body part. Understanding this gradient system leads us to an important realization about human health. Pattern formation is absolutely critical because it explains how complex life is even possible. Without this spatial organization, a fertilized egg would merely divide into a disorganized, chaotic mass of identical cells, much like a tumor. In real-world medicine, errors in pattern formation lead to severe congenital birth defects, such as missing limbs or organs developing on the wrong side of the body. Furthermore, the exact same signaling pathways are often hijacked by cancer cells to spread, making this knowledge vital for oncology and regenerative medicine. Knowing how crucial these signals are, we need a way to visualize them. To understand how cells know their position, we use a mathematical model famously conceptualized as the French flag model by Louis Wolpert. Imagine a chemical signal diffusing away from a source cell into a line of receiving cells. As the chemical travels through the tissue, it gets degraded, so its concentration drops off exponentially the further away it travels. This creates distinct zones of concentration, much like the distinct colored bands of the French flag, which dictate different cellular fates. This conceptual model translates perfectly into a mathematical equation. We can model this steady-state concentration profile using a standard exponential decay function derived from the physics of diffusion. Here is the formula. The variable c of x represents the concentration of the morphogen at a specific distance x from the source. The term C0 represents the maximum concentration right at the source. Finally, the Greek letter lambda represents the decay length, a constant telling us how far the morphogen travels before degrading. If source concentration increases, the signal reaches further. If decay length decreases, the signal breaks down quickly, affecting only nearby cells. Now that we have our formula, let us see this with a concrete example. Let us apply this to a developing fruit fly embryo. A morphogen called bicoid is produced at the anterior end with an initial source concentration of 100 units. The decay length for bicoid in this tissue is 50 micrometers. We know that a cell will develop into head tissue if it receives a concentration of at least 36 units. Our goal is to find out the concentration of bicoid at a distance of exactly 50 micrometers from the source and determine if a cell at this location will become head tissue. To solve this, we will use our exponential decay formula. We need to plug in our known values for the source concentration, the decay length, and the specific distance of the cell in question. Here, we have substituted 50 micrometers for the distance x and 50 micrometers for the decay length lambda into the exponent of our equation. This substitution represents the specific physical location of the cell we are analyzing. By dividing 50 by 50, the fraction in the exponent simplifies neatly to -1. This makes the subsequent calculation much more straightforward to perform. Continuing our calculation, we evaluate the exponential term. The mathematical constant e is Euler's number, approximately 2.718. When we raise this to the power of -1, we get roughly 0.368. This decimal represents the fraction of the original signal that survives at this distance. We multiply our initial source concentration of 100 by this surviving decay factor, giving us 36.8 units. Because 36.8 is strictly greater than the required threshold of 36 units, this cell receives enough signal and will successfully develop into head tissue. That first example showed us how to find concentration. Now, let us look at a slightly more complex scenario involving the French flag model, where a tissue is divided into 3 distinct zones: blue, white, and red. A morphogen is secreted with a source concentration of 200 nanomolar and has a decay length of 100 micrometers. The blue gene is only activated when the morphogen concentration is above 27 nanomolar. We need to calculate the exact physical boundary, meaning the maximum distance x where the tissue stops being blue and transitions into the white zone. In this problem, we are not solving for the concentration because we already know the critical threshold is 27 nanomolar. Instead, we need to work backward algebraically to solve for the physical distance x. We start by substituting our known threshold of 27 on the left side, our source concentration of 200, and our decay length of 100. This sets up the complete equation governing the boundary. Next, we isolate the exponential term by dividing both sides by 200. This gives us a decimal value of 0.135 on the left side. Now that we have isolated the exponential term, we need to bring the variable out of the exponent. The right side contains the base e raised to our unknown exponent. To remove the base e, we must apply a logarithmic function. Specifically, we take the natural logarithm of both sides of the equation. The natural logarithm of 0.135 can be calculated using a scientific calculator, yielding a value of approximately -2.0. We substitute this numerical value into the left side, leaving only the fraction containing our unknown distance on the right. We are now at the final step of our calculation. To isolate and solve for the final distance x, We simply multiply both sides of our equation by -100. -2.0 times -100 gives us exactly 200 micrometers. This means that from the source, all the way up to 200 micrometers, the morphogen concentration remains high enough to activate the blue gene. Exactly at this distance, the concentration drops below the 27 nanomolar threshold, marking the precise physical boundary where the tissue transitions from the blue zone into the white zone. Let us visualize everything we have just calculated. Here, you see the classic French flag model diagram. The vertical axis represents the morphogen concentration, and the horizontal axis is the distance from the source. Notice the solid black line curving downwards? This is the exponential decay gradient we modeled mathematically. The horizontal dashed lines represent the genetic thresholds, like the 27 nanomolar limit we just used. Where the gradient crosses these thresholds, it creates sharp vertical boundaries. These boundaries divide the tissue into distinct territories, shown here as blue, white, and red zones, each destined to become a different part of the organism. We have covered a lot of ground today, so let us review the key takeaways. Pattern formation is the essential biological process that gives a developing embryo its spatial organization and complex body plan. We learned that this is driven by morphogens, which create concentration gradients across tissues. By reading these chemical signals, cells can determine their exact physical location and activate the correct genes. We also saw how this elegant system can be modeled mathematically using exponential decay. Ultimately, this incredible process is what allows a simple cluster of identical cells to reliably transform into a highly structured, fully functional organism.